Our next speaker is Beata Turonova from Max Planck Institute, who will be talking on TANGO, not the dance, but decoding, but the framework for decoding spatial organization in Cryo-ET. The stage is yours.
Thank you. Can you hear me well? Yeah, but I have this one. So, let's try. Okay. But hey, welcome to my talk. I'm very grateful to be here again after all these years. I counted. I will not tell you the number when I was last time on the stage on CCP-EM. This time I would continue on the rich context that we see in the tomograms. And as a bit of a motivation, this is one of the tomograms of T cells that we get. And you see there is a lot of things in it. We can see nuclear envelope, we can see mitochondria, and there is a rich information that we can harvest. And we are actually increasingly getting better in this. So you just saw one of the picking possibilities, Mart's talk yesterday about the segmentations. So we can actually get a lot of our tomograms, right? Not just the one thing that we might be interested, but all the other things. And this beautiful tomogram was put, this video was done by Jan Philip Kreising from our Martin Beck Lab group. And you can see that this actually adds another layer of complexity, right? Because the next challenge that we now have with these kind of things is, how do we analyze such data? Ideally within the context, because if you look at this, it's quite diverse, right? There is a diverse mixture of features and even representations of these features. And ideally, we would like to have some kind of a tool to analyze them that would provide us with one single pipeline and not not very hypothesis-specific tools. And I would like to motivate it a bit with 3 recent studies from our lab. So we are a nuclear pore complex lab, and traditionally we were always picking the nuclear pore complexes by it's actually a pointer works. Yeah, by manual picking, right? And again, we use context here to pick because we don't really see the pore that is here, but we see the hole in the envelope and we know that this is a pore. So what we do is we basically always put 2 points here to define the position and 1 point to the cytoplasmic side to get the orientation. And we pick like this and we already get an average without any alignment that is pretty decent and has the significant features from the NPC. So we see cytoplasmic ring on the top, we see this nuclear envelope on the side, and there is the inner ring inside of this, and there will be nuclear ring on the other side, right? And what we always do then is the alignment, it improves quite nicely. The thing is quite big, so we tend to use the 8-fold symmetry and split it into 8 subunits. We get 8 times more particles, and we continue with our averaging on the subunit level, right? And we can push it even further. So we can even split this one asymmetric subunit into the separate rings. And again, just split it and run focus alignment on those to improve every single part of this. But it's actually very important. What we always also kept doing is we kept the affiliation of all of these sub-asymmetric units of the rings to its pore. So we always know for each of the particles that we have in the data set, to which pore it belongs and where on the pore it sits. And this provides us with a very easy way to make all the kind of cool analysis. We can do more precise circumference, more precise diameter. We can see whether this is really circular or maybe squeezed by measuring the distances of the opposite subunits. We can see how well it aligns by checking these orientations or the inner angles, whether they actually comply with 8-fold symmetry, right? And we can, of course, do it between the rings as well. So we can look between different angles. We look between the rings, what are the angles there, how they're oriented, what are the distances, and again, we can describe local deformations of the pore or the envelope around. And this particular pore comes from Patrick Hoffman, and indeed, the structure, while impressive, was not as impressive as the analysis that came from this, because what he actually in the end studied was how the pore dilates or constricts under osmotic shock. And we are very happy with this pipeline. It's very reliable, because we get very clean particle list. On the other hand, there are some issues that we were battling with. One is obviously, in order to pick manually, we have the substantial part of the pore has to be present. And when we are having a FIB milling and we cut a lot of our data, we often lose some of the parts of the pore, and we would not be able to pick those parts anymore. The other problem is that we actually always impose this 8-fold symmetry by default. And now we know there are more and more cases popping out that it's still outlier. The pore is 8-fold symmetric, but there are some cases when it's not the case, and we are all missing those. We miss those cases when we impose the 8-fold symmetry. And last but not least, obviously, it's manual picking, and nobody wants to do it in the 21st century. So we kind of moved to now and finally figure out or find the proper 3D template matching implementation from Will1 in Stopgap that worked quite well. This is now as a template here, there was, you can see, I hope all of you, these beautiful pores here. This is actually, the template was a cytoplasmic ring asymmetric unit, so it was not the wall subunit. And we can beautifully see them. And if I now color it differently, you see it even better how it stands out, but there's still some background that it would be quite intervening with our analysis. Now, if you look into this particular one and start overlaying with the other template, so now we add the inner ring, we see again, it works very nicely. We get almost all 8 ones, not all. And again, we go for nuclear ring and we have this pore, right? And again, depending on what you want to do in your analysis, for us, it's important to get as much particles as possible. So our pipeline in the end ended up being using rather lower threshold because we see them quite well, but you still have to set a number at which you extract these peaks. So we prefer to have a lower number to get all the pores in. And then we manually clean it, which kind of beats the purpose of having automated particle picking if you have to manually clean it. And most importantly, we still have to go and write extra scripts to other affiliation because we immediately see, our brain immediately put all these points to one pore, right? But in computer, you don't have this information. And if you want to make all this cool analysis on the shapes and all the different outliers that you have there, you have to affiliate it back to the pore, right? And this is where, again, a bit of a manual work and scripting starts. And again, so it's a bit of a work. Jan Philip, however, did it. This is his data now. He was looking into the HIV capsid passing through the nuclear pore complex. So one part of his analysis was to actually identify all the pores. And because he did it actually with template matching, and because the confidence here was quite high, he didn't have to actually run averaging so much. And he could look into individual events of, this is the HIV now segmented passing through the pore. And the first line is a cytoplasmic ring, then it passes through the inner ring, and then it passes to the nuclear ring. And essentially, because you don't have to average and you don't have to impose any symmetry, you see what's going on. And you see in some cases, there is, for instance, 8-fold symmetry here already, but there is still a place. So the pore kind of cracks here. And essentially, one of the outcome of this study, this is like all the events we kind of captured to monitor and see that we, in the end, added some MD simulations to show that most likely, because it's a bit too big to fit without any friction, it actually cracks the pore a bit when it's passing through. And the additional nice analysis that was necessary to be done is then, if it actually kind of squeezes itself through and doesn't rupture, how the lattice looks like, right? So we looked into the various events. We have one mature variant that was actually outside of the cell. We had few capsids in the cytoplasm, some passing through the pore. I'm not sure if you see, but this is an envelope, so this is really inside of the pore, and this is now on the nuclear side, right? And again, we use context to classify, so we do subtomogram averaging on all of these hexamers separately based on when they are. We discover some differences despite the low resolution, and most importantly, he was able to identify the pentamers, and also kind of quantify that this lattice gets a bit disrupted, but we cannot actually say it's physiological, and it is from the passage because we have a worse signal in the nucleus, so it might be that just we are not able to align it so nicely. But anyway, the point here was not to get a high resolution structure of a hexamer. The point here was to describe the changes in the lattices. And my last motivation project for what I want to introduce next is analysis of the chromatin. And again, we got again Jan Philipp, this time in collaboration with Sergio from Gerhard Hummer, that got this nice data of the T cells with plenty and plenty of nucleosomes, and we somehow localized them. Sergio did some prediction on how the linkers could be connecting the nucleosomes. And if you look at this kind of data, what is actually interesting for us would be, okay, is it somehow, is there some pattern in there? Because this seems pretty random, right? Is there some kind of, because are there some conformations that are existent? Because from all the in vitro studies, there were like always reported these stuck nucleosomes, also these triplets in the same orientation, or these helical structures essentially are 3 copies of this on one side, right? And do we have this in our data? And how do we get this answer out? Or maybe there's something completely different in there. And all of these 3 cases kind of show what kind of diversity is there when we want to analyze our data. And it always seems that every project needs a bit of an individual touch, so you have to come up with your own thing to analyze things. We have different inputs, we have the segmentations, we have the particle lists, we can have the scores map or the probability maps or the magnitude maps. The problem here is that the tools are less developed because there is not really, it's very hypothesis driven. You can ask the question, okay, now I have my ribosome and I have my membrane segmentation, how far they are. And then you can write a script that actually measures. And this is the problem because these scripts are always hypothesis driven. They are typically in-house. So even if they are put out there, as was very funnily mentioned in the sketch yesterday, nobody really cares if anyone else can ever run it, as long as they are public, right? And they are tied to the specific pipeline, so the format of your particle list and stuff like this. That's not by default quite reproducible. And all of this makes it actually less accessible to the users, right? If you are a newcomer to the field, if you are not very good in programming, if you do not want to deal with particle lists, this is a bit of a problem, how to actually get the most out of your data. And it was bugging me for a while. And then we kind of started to look for the unifying feature. Is there actually something that all of these things have in common in the sense that we could actually have somehow a toolbox or a pipeline that would not be so diverse as are our data and they would still do the trick? And the answer is yes. We actually have something that unifies because all of these inputs that we can have can be in the end transformed to oriented points. So points in 3D that have actually an orientation. And when we have this representation, we can start looking into different fields. And in computer graphics and computer vision, there is a whole body of research on something called point descriptors. And it deals with the point clouds, which is, again, a set of points in 3D. This is a famous computer graphics bunny, computer vision bunny. And this one, typically these point clouds come from some laser scannings. And then you have these points and you want to connect them in the surface. And there are different tasks you can do with that. But what the point descriptors actually essentially do is they describe each of the point in the point cloud, not by the point itself, but by its neighborhood. So if I have now my point cloud here, I call the one particle of interest or the point of interest, the query point, which is the green one here. I defined area on which I want to evaluate this point, and I call this area support. So this would be this one. And then I can do many, many features. The very simplest one is to count the number of points, right? So this query point has 7 neighbors in this area. If I look into this point, it has only 4 points, right? And this is the very basic thing you can do, but obviously the list of features you can do is endless. You can do the mean distance, median distance, you can do some standard deviation variations, you can do histograms. There's plenty of things you can do. And if you do this, then you essentially end up again with something from the previous talk, a feature vector, right? So you have a vector of features that describes your point by its surroundings. And in computer graphics, this is used for pattern mining, matching, to find similarities between the surfaces. And it's very powerful. And thus we decided to actually bring it to Cryo-ET. And this is Marcus, my PhD student that was brave enough to take up on this, and it's basically his work that I'm presenting. Our point clouds are our particle list or oriented points. So as I said, even if you have a segmentation, you can turn this into the list that we can use. And for us, we don't have only positions. We also have the orientation, which is very powerful because every point now can be actually described by the by the orientation, which is 3 numbers, and position that is also 3 numbers. So this is my query point now, and this is my support that I want to evaluate this point on. The other very powerful thing that we have is because these particles typically are, because they are oriented, we know what is the canonical reference orientation. So before we analyze them, we can all, for each of these particles, we can turn them to the canonical reference frame together with its surroundings. So you see the relative orientations and positions remain the same, but everything kind of turns, which makes this method rotationally invariant, which is, again, very powerful and makes our life way more easier. And if we kind of start with that, we can compute something we call a twist vector. So for this point, for instance, the basic twist vector would be the 3 relative orientations to this point and relative position of this point to the query point. This would be the third line. And equivalently for this point, it would be this line. And for this point, it would be this line, right? Now we move to the other point. Again, imagine that we rotate it to the canonical reference. And we will get a twist vector that has 2 lines only because we have only these 2 points in the support, right? So we do this and we compute a twist vector for all of the points that we have in our, or all of the particles that we have. But as I said, this is a basic. We can add endless number of features and we provide some basic 20 of them, and we call that twist descriptor that apart from this relative position and orientation have some additional deviations from axis and stuff like this as a features. And if we now have this as a framework, we can actually have a unified pipeline to perform all the analysis that I've showed you before. For a brief overview, so we start with the particle list. We can do some basic analysis of this particle list. You run a nearest neighbor analysis to find out what is the average distance to your particles. So you can have a bit more informed idea how large the radius of your first support will be. But if you know it already because you work with HIV and you know what are the distances, you don't have to do it. You can go directly straight to setting up your support. Then you compute this twist descriptor. You can customize it further. You can say, I want more feature, I want less feature. I want a support that is smaller. I want to have a bit more focus area to look into. And then you perform an analysis. So, and the analysis can be, K-mean clustering, can be polar histograms. In this particular case, you see that there are 2 orientations that are very prominent, and almost every particle has a neighboring particle with a very similar orientation, right? And when you know this, you can see, okay, is this, and you can do it directly. Again, the customization is voluntarily, but once you have this, you can decide, am I done? Is this all I want to know? Or maybe now I know that there are these orientations. Maybe I want to add one more feature. I want to add one more feature to the vector that looks into the angles between these neighboring things. So I orient them and look into the angles, right? And then I get a different type of clustering that maybe points to something interesting in my data. And this might sound a bit vague, but I'll go back now to all the examples I showed you before. So let's start with this particle list coming from the scores maps of the NPCs that we have the subunits, and you can see if we have a low threshold to have all the particles, we have a lot of junk, right? And I've blown up here based on the annotated ground truth from Jan Philip, what we know or we believe it's poor, and what is actually junk we want to clean. And again, the point was not just to clean, but we also wanted to get this affiliation and get these points clustered together. There we start with a bit of a trick, because we know that we can also move the particles within their reference frame by their internal orientation. So we can shift them along x-axis, which brings all these things that are actually belonging to a ring more together, while the others wander randomly off. And then we set out our support base. So ideally, so for each query point, the wall pore could be actually in it. Then we compute a twist vector. We can say, okay, maybe this thing is cylindrical. I don't need a spherical support. I kind of reduce a bit and change the support to the cylinder. And then I do proximity clustering. And this is not only proximity in terms of Euclidean distance, but also angular distance. So these things that are clustered together have to be close in the space, but also have to have a similar orientation. And if I do it, I get quite a good outcome. So let's zoom in here. You see it's not perfect. For instance, there is some false positives here. Something is missing. Here, on the other hand, there is one point that was added that was missed here and here as well, right? And again, this was missed here or this was missed. So it's not perfect. Any automation will not be as good as your manual work, obviously, right? But if you have 200 tomograms, this is very good. The other example that we got was the HIV capsid, right? So again, we have a particle list. This time we have, there are pentamers in here. Like you can see, this one is a pentamer, but this is like just a list that we know there are, most of them are hexamers, some of them are pentamers. And again, this time we do simple nearest neighbor analysis to establish the distances between our query point and the first shell. If you want, you can do 2 shells, depends on what you are after. You define the support based on this radius, you compute it with descriptor. And again, this thing is locally flat. Maybe it makes sense to use cylinder instead of the sphere if you want. But this is now a lattice. And lattice, there are a lot of geometrical features you can compute on. And one of them is so-called alpha complex. So I can order the points that I have in my surroundings and measure, for instance, the inner angles and use this as a feature of my descriptor. And if I do it, I can then do PCA analysis and k-means clustering, and I get the assignment that actually assigns my pentamers. And again, not perfect. This is like 1 or 2 missing. Sometimes it does the good job. Sometimes there is a false positive. Again, not perfect. If you would have 10 capsids, you are better off doing it manually. But again, for large data sets, this was pretty good, and it gives you at least a good start where you can start looking into the things. And the other thing that kind of came to mind when we were working with the hexamers was the symmetry, right? Because if you think about C symmetry or some more symmetric or some platonic solids, then these things are misaligned just by a bit, right? But because the orientation in the in-plane can be assigned arbitrarily based on the symmetry, you might get one orientation here and the other one here. So of course you cannot just do simple distance or angular distance. And you can even not do the simple modulo based on the symmetry because it will not work in all the cases. So Marcus came up with the angular score that essentially if everything is perfectly aligned between you two of your particles, the score is 1. And if you go as far as you can get, it's 0. Well, because if you go here, again, you're returning to the next vertex. It works also in 3D for the platonic solids, not just for C3 symmetry. And it's very useful because you can, for instance, use it to estimate what is the prevalent symmetry within your lattice. See, so we would expect here it to be 6, and it is, and it's statistically significant here. But if you have irregular lattice, if you are not sure if there is anything or any regularity in your lattice, this is very helpful to actually give you an overview. And because this additional feature in the twist descriptor, you can also just simply cluster based on it. And this is now also HIV. This is a VLP of immature HIV particle. And you see the ones that are really good have all 6 neighbors and are nice. But there are some that are in those cracks that we always have in those VLPs. And you can either throw them away because they will probably worsen up your average if you are up to high resolution, but you can also just look into them and see, oh, this guy actually doesn't have 6, but only 5 neighbors, although there should not be any pentamers in there, right? So it can point to interesting parts of your data. And last but not least, I go for the chromatin. So for this one, we actually created a synthetic ground truth because we wanted to have a bit more quantitative results on this. So we put all of these 3 constellations in there. You can see it here, and plus all these single nucleosomes that are orange, right? And again, we do the same thing. Particle list, some nearest neighbor analysis to estimate the support, and to do the twist descriptor. And now with that, there are multiple approaches you can take. One is, I know what pattern I'm looking for. So I look for the stacks. Now this is my query point. I put my support around this, so it's centered around the query point, and it extends only up. So I actually look only here towards in all my particles. And I know that these angles between this and this should be almost similar in plane. So I just plot this, and actually this one gives me more than 99% of my particles out from the data, right? Or I know that I want to look for the triplet. So again, I can set the cone to look only here and see, is there something in there? And is there some distinct relative orientation? And again, I do this is the polar histogram you saw before, and you clearly see that these 2 peaks are actually corresponding to these 2 these two nucleosomes. And you can basically just get this out and average this, and in both cases get average without any classification. And I can also look for these helical arrangements, where I basically do clustering on this metric versus these angles that I picked for this rotational direction, and I can various clusters that describes different arrangements, right? So this is nice, but of course, way more powerful is to have something more agnostic. What if I do not know what patterns are there? And again, you already have the twist descriptor, so you can just do PCA analysis on all the features. You can maybe think of additional features that might make sense to add. And when you do this, you can also run some statistics, and it tells you, okay, these three features are the most prominent. They actually explain 95% of variance of your data. So you do clustering based on these three features, and then you get, I don't know how many you ask for clusters, and you can look into them. You can create an average, you can go back to tomograms and see how this looks like. You can then go with those colors back to your particles and see what's actually going on in there. And essentially, these were like completely 3 or 4 different types of analysis that were all done within one framework that we very poetically call TANGO, which stands for Twist-Aware Neighborhoods for Geometric Organizations. And we hope it provides a bit more holistic or agnostic tool for these contextual analysis, that you don't have to always think about how to do things, and we can start with that. It's Python-based and obviously fully open source. We try to have it software agnostic, so we provide support for all the things that are usually out there, but if you have issues with your inputs, just shoot us an email, we will try to do our best. It provides the twist descriptor, additional features, some descriptors that are more suitable for lattices, some descriptors that are based more on counting and histograms. There are some filters that are there. You can combine them as much as you want. But the best thing is it actually allows you to add your own features. So whenever you think, okay, there will be a feature that I want and it's not there, if you follow the programming logic of the Python module that we have, you can add your own features, you can add your wall descriptors, and you can use them. And even better thing is that it has a GUI. So you can actually look into your particle list, visualize it, you can play with filters, you can get a bit more feeling of what is in your data. And again, this is already automated, and it's automatically generated based on the Python module. So if you add your features to your Python module, it will automatically also appear in the GUI without you having to do anything. Okay, and that's almost it from my side. What I want to still say is that, going back to this beautiful image, I only showed you, analysis of nucleosomes, analysis of hexamers, but obviously you can take any 2 particle lists. You can take your ribosomes and you can take your segmentation of a membrane and analyze them together. So you don't have to analyze only one particle set. You can mix those based on what you have in your data. And with that, I would like to thank my team. So we are shrinking a bit. So this is the current team. These are already alumni. Had a lot of fun. We are embedded in Martin's Beck group. I'm hiring, so I have a PhD position from March on, in case anyone is interested in a bit of a development. And yeah, if you want to read more, the manuscript is out. It's all part of the Cryo-ET, which is a bit more generic, larger toolbox for contextual analysis in general. And Marcus even make a nice tutorial video for the how to use the GUI if you want. And with that, I'm happy to take the questions.